Hello, everyone. We're going to get started. We are very excited to have this joint AII RCMB webinar titled Preclinical Models in COPD, which is the best mouse? We are very fortunate to have three speakers with expertise in murine models of COPD who will each describe a particular model. At the end, we'll have a group discussion to weigh the benefits and drawbacks to each mouse model and consider what situations may be best for particular models. Please feel free to put questions for the speakers in the chat. We will answer all questions at the end of the talk, and we will not answer questions in between speakers. And then keep in mind the second half of the webinar will include a group discussion with pre-planned questions and then also the questions from the audience. Wonderful. And then if Dr. Curtis, would you like to introduce our speakers?
Yeah, I'd be happy to. So I want to apologize in advance to everyone for my voice. I have a grandchild-induced upper respiratory infection, and I might be off mic coughing between things. And I also want to thank Amy, who did the overwhelming bulk of the work for this. She's been fantastic to work with. So we'll dive right in. Welcome, Bradley. Great to see you could make it. So our first speaker is going to actually be Dr. Uma Sajjan from the Lewis Katz School of Medicine at Temple, where she's an associate professor in microimmunology and inflammation, and also in thoracic medicine and surgery, and biomedical education and data science. And Uma's going to talk about the elastase plus minus LPS model. Over to you.
Okay. I would like to take this opportunity to thank Dr. Amy Attaway and Jeff Curtis for organizing this webinar. Also, I thank them for giving me an opportunity to present our Elastase LPS model for COPD. Next slide, please. COPD patients often show lung inflammation, emphysema, chronic bronchitis. Therefore, it's important while developing a model, it's very important to consider what aspects of the disease is one interested in. Is it lung inflammation, chronic bronchitis, emphysema, all the features? If the intention is to study development of disease, one can expose mice to known risk factors of COPD, such as cigarette smoke, particulate material, or others, and examine the changes that occurs in the lung. If the intention is to examine progression of lung disease following acute exacerbations, one may consider using the molecules that are increased in the disease conditions along with the known risk factors of COPD. This will augment the development of COPD features and allows to investigate the effect of acute exacerbations. Next slide, please. We were interested in examining the contribution of rhinovirus in the progression of lung disease in COPD. Therefore, we developed a mouse model which shows moderate to severe COPD-like lung disease, including lung and airway inflammation, emphysema, and remodeled airway epithelium. It's known that protease and antiproteinase ratio is increased in COPD. And it's thought that this increased ratio of protease to antiproteinase contributes to disease development. The proteases that are particularly increased in COPD lungs are matrix metalloproteinases and elastase. So we use the elastase, one of the endogenous factor. Why did we choose LPS? Because it's present in abundant amounts in cigarette smoke and other risk factors. So both elastase and LPS has been individually used to model COPD-like lung disease. But the drawback is treatment with elastase alone causes extensive emphysema with mild inflammation, whereas treatment with LPS alone causes airway inflammation with no changes in alveoli. I'm going to show you some of these results when I'm presenting the actual results. So to create a model of COPD, we combined these two agents to induce COPD-like lung disease within a short period of time. Next slide, please. The method that I'm going to show you will not require any special equipment and can be done in all the labs. In our experiments, we used 8 to 10 weeks old C57 black 6 mice, and mice were treated intranasally with elastase on day 1 of the week and LPS on day 4 of the week for 4 weeks. We used 1.2 units of porcine elastase and 7 micrograms of E. coli LPS. It's very important to choose the LPS that does not cause too much of an inflammation. So that's why we used an E. coli LPS. After the last treatment, mice were allowed to rest for 10 days, and then we examined for inflammatory and morphological changes in the lungs. Next slide, please. In this slide, I've shown H&E stained lung sections for all 4 groups of mice. As you can see here, the first panel on the leftmost panel, upper leftmost, shows the mice that are treated with PBS, which doesn't show any inflammation. And the next panel, which says Elastase C and D, shows mild inflammation, mild alveolar inflammation, and also show accumulation of macrophages in the alveolar region. On the other hand, LPS treated mice show peribronchial inflammation. Mostly the inflammatory infiltrated cells were in neutrophils. In contrast to this elastase and LPS alone treated mice, the mice that are treated with combination of elastase and LPS show extensive inflammation in both peribronchial area and alveolar area. And you can also appreciate the accumulation of macrophages in the alveolar space and also neutrophil accumulation in the peribronchial space. Unfortunately, I can't point out, but I can only explain. Next slide, please. So we then quantified the lung inflammation by measuring cytokine levels in the lung homogenate supernatants. Compared to PBS control, all 3 groups of mice showed significant increases in IL-1 beta, IL-6, TNF alpha, and MIP2. But these cytokines were significantly higher in elastase LPS group compared to LPS or elastase treated mice. Then we also quantified total and differential cells in the lavage fluid. We found that in all 3 groups, that's LPS, elastase, or LPS, mice treated with elastase and LPS showed increased number of total cell counts in the lavage fluid and also increase in macrophage numbers and neutrophil and lymphocytes. But what is interesting is elastase LPS treated mice show much more higher total cell counts, and also macrophages. Then we conducted flow cytometry in collaboration with Dr. Curtis, and we quantified T cells, B cells, as well as different type of macrophages in the lungs. All 3 groups of mice again showed increased CD4 and CD8 positive cells, but it was much higher in LPS treated mice compared to elastase alone or combination of elastase and LPS groups. Coming to macrophages, we differentiated this different populations of macrophages based on the expression of CD11B and CD11C. Double positive, all 3 groups of mice showed double positives, increase in double positive cells, as well as CD11B positive and CD11C negative monocytes. We call that CD11B positive and CD11C positive macrophages as atypical macrophages, and our ongoing studies suggest that they are mostly have inflammatory phenotype. But there was not much of a difference between the groups when we looked at the macrophage population. And we then measured the MPO activity, which actually signifies activation of the neutrophils in the lungs. And again, LPS treated, as expected, LPS treated mice showed increased MPO activity, that indicating that there is a increased neutrophil activity, but it was much higher in elastase LPS group. And elastase alone treated mice did not show any MPO activity, indicating that there was no neutrophil activation in those mice. And remember that we did not even see a lot of neutrophils in this elastase LPS treated mice. I mean, elastase only treated mice. Next slide, please. So one thing to remember is COPD patients also show small airway remodeling with goblet cell metaplasia. Therefore, we wanted to test our model whether this model also shows airway remodeling. First of all, we wanted to find out whether this might show goblet cell metaplasia. So we stained the sections with periodic Schiff's reagent. When we use this stain, the goblet cells, those are mucin-producing cells, will stain pink. So as you can see here, the control mice that are treated with PBS does not show any goblet cells either in the large airway or the small airways. But other three groups actually show goblet cells in both large airways and small airways, which are represented by arrows. One thing to notice here is Elastase LPS treated mice show a lot more goblet cells in both large airway and small airway. Another important feature that we observed was epithelial hyperplasia in the small airways and also thickening up the airway wall. So this was very interesting to us because COPD patients do have these features. Their small airways have goblet cells, goblet cell metaplasia, and also they show thickening of the airway wall. So we then quantified the airway wall thickening by morphometry, and we showed that it was only increased in mice that were treated with combination of elastase and LPS. Next slide, please. So how about emphysema? So as expected, LPS and PBS treated mice had no emphysematous changes, that is degradation of the alveolar septa leading to enlargement of the airway alveolar sac size. So elastase treated mice show mild to moderate emphysema. As you can see, those are depicted by asterisks. But in mice that are treated with a combination actually show larger airway sacs, indicating that there was extensive damage to alveolar septa. And we quantified the emphysema by measuring the card length. And again, there was emphysema in both elastase and LPS, elastase LPS treated mice, but it was much higher in the mice that are treated with combination of elastase and LPS. And accordingly, the total lung volume also volume, which is another measure of emphysema, was also much significantly higher in mice treated with combination of elastase and LPS. Next slide, please. So what are the implications? Like, does the respiratory mechanics change when there is when these mice have emphysema? So we did some respiratory mechanics using CIREC Flexivent machine, and we found that these mice show increased compliance and reduced elastance, meaning they, just like a COPD patients, they have a lot more compliance. The lungs can expand easily, but they cannot recoil back to the original state. So that is decreased elastance. So these mice actually recapitulates those functions. Next, so in summary, these results suggest that moderate to severe COPD like lung disease occurs in mice treated with combination of elastase LPS treated mice, and this model can be developed in a short period of time with no special equipment. Next slide, please. So having this model, we wanted to see, remember initially I told you our intention was to examine whether the after acute exacerbation, whether the lung disease progresses in COPD patients. So we just wanted to model that. So we wanted to see whether this model is suitable. So we used the rhinovirus because to cause as an acute exacerbating agent, because rhinovirus is a respiratory virus and it causes self-limiting upper respiratory infection in normal subjects. But in COPD patients, it causes lower airway infection leading to acute exacerbations. And rhinovirus is associated with accelerated decline in COPD patients. So to investigate whether the elastase LPS model is suitable for studying the effect of rhinovirus infection, we infected the mice with rhinovirus by intranasal route and examined for viral load and distribution of virus in the lungs and cytokine levels. Next slide, please. We measured the lung viral load by determining the levels of viral RNA by quantitative PCR. While PBS-treated mice cleared the virus by day 7 almost completely, elastase LPS mice showed virus up to 14 days post-infection. At all the time points, elastase LPS group showed higher viral load than PBS group. Rhinovirus, unlike influenza, it primarily infects airway epithelium. So here we can show, we are showing the immunolocalization of virus to see what kind of cells are infected. So at 1 day post-infection, we can see virus, the bright green color is the virus infected cells. So both control and elastase LPS treated mice show virus in the airway epithelium, but more number of epithelial cells were actually infected with virus in elastase LPS treated mice, indicating that they're more susceptible to infection with rhinovirus. Even after 14 days post-infection, we could see some virus in some of the airway epithelium, indicating that this virus replicates within the airway epithelium of the lungs and persists. Next slide, please. We then examined whether increased viral load increases inflammation. We measured TNF alpha and IL-13 protein levels in the lung homogenate supernatants of these mice. And we found that levels of both TNF alpha and IL-13 is significantly increased in elastase LPS treated mice infected with rhinovirus. And I just want to point out that when COPD patients were experimentally infected with rhinovirus, these patients actually produced more sputum, meaning they had expressed more mucin. And IL-13 stimulates mucin expression, so these mice also show a lot more mucin gene expression. So in summary, next slide, please. Elastase LPS treated mice show both emphysema and chronic bronchitis phenotype. These mice show some structural and molecular features similar to that observed in COPD patients, just such as airway wall thickening, goblet cell metaplasia and overexpression of mucin genes, accumulation of macrophages, T cells, and neutrophils, and they're more susceptible to rhinovirus infection, and they also show exaggerated prolonged responses to rhinovirus infection. So I wrap up this. Thank you.
Very good. Yeah, we're trying to save as much time for discussion at the end. So our next speaker is Dr. Francesca Polverino, who is the Lester and Sue Smith Professor of Medicine at Baylor College of Medicine in Houston, Texas. Francie?
Thank you, Jeff. Thank you, Amy. So I was told that I'm allowed 8 minutes, so I'm going to stick to this 8 minutes so that we can have a very nice, lovely conversation. So I'm going to talk about the most common model of COPD, which is the smoke model of COPD. And this is going to become at the end a pro and con debate because I'm very much pro the smoke model, even though I know that a lot of you hate it because it's a long model, but for me, it's the best model. So I'm going to talk about the factors to be considered when you pick a model versus another model of smoke, because there are several models of smoke, and we're talking about the black 6 mice, which are the most common mice, because for the bulb C, they're a little bit more susceptible to smoke, so the timing might vary a little bit. So first of all, is the type of the exposure. The nose only is literally each mouse is restrained in a small chamber and receive the smoke right on his nose. It's a very aggressive way of getting the smoke to the mice, but it requires a shorter exposure. But you need to monitor the mice very tightly because the mice sometimes when they get a nose only, they can seizure, they can die. So, and you can also expose a fewer number of animals just because each one of the animals requires his own chamber, his own little space where you put the cigarette. Then you've got the whole body exposure where you put a chamber, where you put a whole bunch of mice. It's a less aggressive way to expose the mice to smoke, but it also requires a longer exposure because it's a kind of mix of mainstream and side stream cigarette smoke exposure. The advantage is that you can expose more animals and it resembles very tightly the pathologies that occur in the human smokers with COPD. And the second factor is the length of the exposure, because each timeline brings to different readouts of COPD. For example, we're talking again about, let's assume that we're having a whole body exposure. You get 2 weeks to 1 month of exposure. You can get acute inflammation, which is mainly innate immune inflammation. You can get some kind of mucous metaplasia, certainly oxidative stress, and apoptosis, mainly of alveolar and bronchial epithelial cells. Then when we go to 2 to 3 months of smoke exposure, which is called subacute exposure, assuming that the chronic exposure is from 4 to 6 months, you get inflammation. You start seeing cells of the adaptive immune system like lymphocytes. You can get some innate lymphocytes and some T cells and some B cells. You can get certainly mucus metaplasia and mild small airway remodeling, oxidative stress and apoptosis. But if you measure lung function at this time in a wild time, you still don't see mostly any change in the lung function. And it's only when you have exposed the mice for long enough to 4 to 6 months of chronic exposure, although Phil Ashbrook, for example, as a shorter model, but let's say most of us, for a chronic exposure, we called from 4 to 6 months of exposure, then you can get really the nice inflammation that you get in the human lung with COPD, with the lymphoid follicles. You get mucus metaplasia, overt smaller remodeling, oxidative stress, apoptosis, that only after chronic exposure you get airspace enlargement. So there are several models. This is the classical model that, especially in US, almost everybody worked on. This is called the TIG, and I'm sorry for the low quality of the picture, but the website of the TIG right now is down. So this is the only picture I could download. There are several models. This is called the TE10, which is the most common. On the left, you see the motor, and then you have these two big chambers where you can put up to 200 to 300 mice. When I was at Harvard, we were smoking up to 300 mice at the time. So this is great for a core, for example. And this is another model of the TIG. It's called the TE2, which is, I think is obsolete right now. I don't even know if they sell it anymore, which is like a smaller machine where you can host a less number of mice, fewer mice, but certainly the TIG is the most common model, at least years ago. Years ago, before the CIREC came up with, another downside of the TIG is that it has a very, very busy customer service. The machine is tends to break very often, and their customer service is not ideal, meaning that you can have a few weeks to fix the machine. So keep that in mind. And then the CIREC came out. The CIREC, which is the same company that makes the Flex event, came up with these two models. One is for nose only, and another one is for whole body chamber. The nose only can host up to 12 mice. The whole body chamber can host up to 24 mice. And again, they are the pros and cons of the nose only versus the whole body. The cons is that it's an excellent customer service. If the machine breaks, you can get it fixed within 48 hours, and you have the automatic records of the smoke exposure, but you can host a fewer mice. So probably this is not ideal for a core. And we have our own. We made our own, because the other machines we're talking about $50,000 to $100,000 for a machine. We made our own. So we basically, with a tube with an exhausting chamber and with a series of plastic tubes, we made our own chamber that looks like this. You put the cigarette right there. So if you have an engineer in your group, you can make your own. You can make your own. You put the mice in there. This is how the mice look. The cigarette gets placed in that tube, and the mice get smoked. And this is a kind of a TIG. And this is our machine that we have in the lab. So you can make your own. It's cheaper. The only thing that you have to make sure is that you measure the total particulate matter. You buy a Cambridge filter, which is a filter that you have in the middle, and they sell the machine. This is a small machine where you put the filter before the exposure, you weigh the filter, you expose the mice, and then you weigh the filter after the exposure, and the difference in the weight of the filter will tell you what is the number of the total particulate matter of your exposure, which basically tells you how much smoke the mice have been exposed to. That you have to be consistent with the number, because if the number is too high, the mice might seizure and might die. If the number is too low, you might have no pathologies. This is the last slide. The COPD-like pathologies induced by each model. So Uma already talked about that. The cigarette smoke causes a lot of fibrosis, a lot of tissue remodeling, a lot of adaptive and innate immune inflammation, and a lot of mucus hypersecretion and alveolar destruction. So you can tell me whatever you want, but the most complete model that resembles all the pathology that you observe in COPD is actually the cigarette smoke model that people don't want to use because it takes 6 months, but this is the most complete model. The elastase does cause alveolar destruction, causes a little bit of inflammation, but it doesn't resemble what you get with cigarette smoke. Then you've got infection that, as Uma showed, that does cause inflammation. It can cause a mucus hypersecretion, but infection itself doesn't really cause alveolar destruction or fibrosis. So what I want to point out is that only chronic cigarette smoke exposure induces adaptive immune infiltration that resembles very much to one that you observe in human COPD, which consists of a B cell aggregating lymphoid follicles and all the rest of stuff. So my conclusion is that all the models are acceptable, and we will talk about that in the conversation that we're going to have at the end, but it all depends on your research question. Thank you.
Well, thank you for being concise and definitive, Francie. We assumed we're suspecting you had an opinion, but so our final presenter is Dr. Bradley Richmond, who's an assistant professor in the pulmonary critical care and allergy division at Vanderbilt University in VA Nashville. Bradley, over to you.
Well, thank you, Dr. Curtis, and thank you so much for letting me be involved in, I think, really something I was thinking we should have done a long time ago when I got this invitation. So next slide, please. So I want to start with a bit of a scientific disclaimer, and that is that mice recapitulate some, but not all aspects of human lung structure and function. And I think this is particularly important when we're thinking about genetic studies because with the advent of CRISPR-based technologies, the activation energy to try to do genetic manipulation of other species has really come down quite a bit and is increasingly feasible. I think that said, mice remain a workhorse. Care is straightforward. There's a lot of transgenic lines that are already available, as well as the reagents needed to study them. Next slide, please. So why might you choose a genetic COPD model? And really, I think the main reason is specificity. It really allows you to perform precise study of the effects of a specific mutation, and depending on your construct, potentially in a specific cell or tissue. Generally, the results are highly reproducible with modern inbred strains, although there is important extrachromosomal variability with things like the microbiome or particular pathogens that may get in a mouse colony. You can certainly combine genetic models with various exposures. We've used cigarette smoke. We've used non-typable Haemophilus influenza challenge. So there's nothing that precludes you of looking at gene by environment interactions. And I think the last thing is that genetic models are fun because they're unpredictable in both good and bad ways. And I'll highlight that in a bit here. The figure to the right just really shows kind of the paradigm that I have of how I think about genetic models, where we typically start by making an observation in COPD cells or tissues. We make a genetic manipulation based on that observation, plus or minus exposures. We then do phenotyping, which includes, do the mice survive? An important caveat. And does the mutation recapitulate human COPD? And then more often than not, we come up with some other discovery that drives renewed hypotheses, and the cycle starts anew. Next slide. So some considerations when choosing a genetic model. Remember, there's genetic heterogeneity among the inbred strains. And so if you're going to be combining your model with something like cigarette smoke, it's important to remember that strain susceptibility can differ to things like cigarette smoke. In fact, there are some strains that have naturally occurring mutations that have been mapped to things like alpha-1 antitrypsin. So it's really important to think about what background you're going to use. You need to consider, do you want a loss of function versus a gain of function mutation? Are you going to introduce that mutation globally or in a specific tissue or cell type? Is it going to be constitutive, meaning present during development, or is it inducible at some time later in adulthood? Depends on your phenotype. You want to study, is it your hypothesis that this model is going to be protective or susceptible? And do you want to just look at spontaneous phenotypes, or do you want to combine this with some kind of an exposure? Next slide. This is a very highly unscientific subsampling of COPD mouse studies that I sort of have seen over the years. And I apologize if your favorite model is not included here. If there's any that catch your eye, the PMID numbers are there in italics. But I just wanted to point out that most of our models are loss of function and most of our models are constitutive. And within that group, there's really a huge number of things that you can search the literature to see if it might be relevant to a particular pathway that you're studying. There are examples of gain of function and some inducible, but they're a little bit more rare. Next slide, please. So I'm going to just kind of highlight some of these points with two mouse models that we've used. Nothing particularly special. I'm not advocating that these are necessarily the best models for COPD, but I think it highlights some important lessons. Next slide. The first model was based, as I said, upon an observation we made in human lung tissue, which is that in the small airways of patients with COPD, we observe a loss of secretory IgA on the airway surface. That's particularly true in airways that have extensive goblet cell hyperplasia or squamous metaplasia. And this was described now several years ago by my colleague here at Vanderbilt, Dr. Vasily Polasukin. Next slide. To understand whether this actually had any sort of mechanistic implications in COPD, we knocked out, can you press next slide, please? I think, yeah, there we go. We knocked out the polymeric immunoglobulin receptor. This was a global knockout, so there was a loss of IgA at all mucosal surfaces, which is an important caveat for this work. But you can see in that figure to the right, that there's a complete absence of IgA in these mice. Next slide. We then performed phenotyping over the course of several years and found that these mice develop a very COPD-like phenotype, which includes emphysema and small airway wall thickening and fibrosis. These findings were not present in young adult mice, suggesting they weren't developmental defects, an important thing to address in your models, but got worse over time. And it really resulted to inflammation, which included both innate immune cell accumulation like neutrophils with protease-antiprotease imbalance, as well as the mice aged into older adulthood, adaptive immune activation with increased numbers of apoptotic cells, both in the epithelial compartment and in the endothelial compartment, all of which are phenotypes that to varying extents have been described in patients with COPD. Next slide. But the most fun thing about this model was really what we discovered that we weren't expecting, which is when we began doing some phenotyping of these mice by flow cytometry, we found that they had an increased number of monocyte-derived dendritic cells. We then used mass cytometry to look back in human lungs from patients with COPD, and we found that they too had increased numbers of monocyte-derived dendritic cells, which we would have, at least I would have never guessed necessarily going in this model. So it just shows you how you can learn a really interesting biology when you use a model like this. Next slide. Second model is going to be set up similar, but is more included as a cautionary tale. So another observation in human airways is that the airways are often deficient in P73 in COPD. P73 is a transcription factor that's been shown to be required for the early stages of ciliated cell differentiation. And we found that there was really a marked reduction of these cells in COPD airways. Next slide. Here, rather than a global knockout, we made a tissue-specific knockout using the Sonic the Hedgehog Cree, which knocks out most of P73, specifically in the respiratory epithelium. The cautionary tale wasn't published, but is that this was the fourth Cree that retried. The first one was mostly fatal. The second one had no phenotype, and the third one, the mice were infertile. So persistence pays off, but I do provide this as a cautionary tale that things don't always go swimmingly. However, in the end, we were able to delete ciliated cells, which you can see there with absence of alpha-tubulin staining and absence of cilia on EM. Next slide. And we found that these mice spontaneously developed neutrophilic inflammation. It was relatively specific to neutrophils, at least at the time points we examined. And these mice developed emphysema as well. Next slide. However, again, we came up with an unexpected phenotype as we were phenotyping these mice. In addition to loss of ciliated cells, which we certainly expected, we found that these mice had an exaggerated and age-dependent decrease in secretory cells. And in this case, this wasn't a discovery. This was a rediscovery of multiple papers, including one by Dr. Polverino's group, which showed that there's a reduction in secretory cells in patients with COPD. Well known and something that we would have never necessarily thought could be introduced by loss of the ciliated cell-specific transcription factor. Next slide. So in summary, mice provide a genetically tractable tool to better understand very specific and targeted questions related to COPD. They may not be as useful for some phenotypes. I'm thinking particularly about phenotypes related to terminal bronchioles, which mice don't have, or basal cells, which mice lack except in the trachea and upper divisions of the main stem bronchi. Genetic studies are fun because they're often hypothesis generating in addition to hypothesis confirming. And remember that transgenic models are easily combined with exposures to study gene by environment interactions, which of course are so important in COPD. Thank you.
Wonderful. Back to you, Amy.
Oh, that was wonderful. Thank you so much I was so excited to hear these speakers kind of give their take on each model. So in this portion of the discussion, we are opening up for questions from the audience, but there were some pre-planned questions we wanted to discuss about the models. And so this was our first one we wanted to discuss is what situations would you choose a certain mouse model over another? I think some of our speakers already gave some examples, but would our speakers like to mention a few situations?
I can start. So as I said in my last slide, it all depends on the question. Sometimes I review papers and grants when they are like, this is a COPD model, and then it's an infection, or it's LPS only. And so, human with COPD. So if the purpose is just to generate emphysema or smaller remodeling, sure, do whatever you want. But if the purpose is to find a treatment for a disease, we gotta resemble how that disease is generated. And in the human, people get COPD by inhaling substances. So it can be cigarette smoke or can be wood smoke, but the toxicant has to come from the lung. Because the fact that the pathology is the same, you get emphysema with other models, doesn't mean that it doesn't mean that the pathogenesis is the same. So what I'm saying is that it all depends on the question. If you want to have a model of emphysema or a model of airspace enlargement, sure, go ahead and use elastase, use whatever you want that creates holes in your lungs. But if you want to call it a model of COPD, you got to make sure that the genesis of the pathology is the same as in humans. That's my 2 cents.
Yeah, I mean, I think the initiation of doing a genetic study in mouse is not terrible, but it's certainly not free. And so you do have to sort of have some sort of a hypothesis, usually based on a human observation of what you feel like that model should look like. So that does sort of require that you have that level of work. I think, I would tend to gravitate probably a little more towards a genetic model over something like a cigarette smoke or an infectious model. If I really wanted to study a compartmentalized response, meaning to knock out X thing in a specific tissue, for instance, if you were interested in COPD related cachexia and you wanted to be able to study how that works in muscle, you could perform muscle specific knockouts versus a global knockout where you'd be introducing, that mutation in the lungs and muscles similarly. So, I think those would be a couple kind of first order comments.
So my take view is like, yes, cigarette smoke model is perfect. I mean, it's not very perfect. It takes a long time, but it does generate some COPD like lung disease. But the question is like, not all the laboratories are equipped with cigarette smoking machine. In Europe, actually, they don't, the IACUC does not allow investigators to expose mice to cigarette smoke. So we have to have some sort of a model. So that's the reason maybe using elastase or genetic mouse models or a combination of certain things like what's found in risk factors, for example, cigarettes, there's abundant amounts of LPS. So using those combinations probably will provide a model. Again, it depends upon what kind of question that you're that are trying to answer. Yes, for development of disease, elastase or LPS model may not be, or a genetic model may not be very suitable. But if you're trying to figure out what happens when you give the infection, what happens after acute exacerbations, like if you infect the mice and what happens, how do these mice actually respond? In our model, we show that it actually responds very similar to human, because in Europe, in England, they actually showed experimental rhinovirus infection causes more sputum production, more IL-8 production, and they also have severe respiratory symptoms compared to normal subjects. All this can be recapitulated with these mice. So yes, I do agree with you, but we have to take everything into consideration when we're selecting a model.
So Amy, we have a number of questions in the chat that actually kind of address this. Do you want to try to go over some of these?
Yeah. Why don't we so let's see. Okay, so we have
Dr. Abman has asked basically what sort of models could be used to study pulmonary vascular disease within COPD, pluses and minuses, and do any of them progress to pulmonary hypertension? Are any of the...
I can start it. So yeah, so this is Steve. So So this is a great question. And I know that Steve is interested in bronchopulmonary dysplasia and in the vascular pathologies that connect BPD with the early origins of COPD. So this question is very relevant. So, you know, the cigarette smoke itself, it causes very mild pulmonary hypertension with some vascular remodeling and inflammation, although in guinea pigs, it seems to have a bigger effect on vessels. Then you've got other models like smoke and hypoxia, for example, that gives more pulmonary hypertension feature and vascular fibrosis. And it mimics a little bit the hypoxemic COPD, pulmonary hypertension. Then in Elastase also, you have got the rapid emphysema with vascular remodeling, but there is very much less inflammation and mild pulmonary hypertension. Then you've got, again, you can combine Elastase with smoke, or you can do Sajjan hypoxia with smoke. But in mice, I have to say, the mice are not the great animals to study of vasculature when it comes to resembling the pulmonary hypertension in COPD. I would say the larger models, like primates, they have a much more abundant vascular inflammation. Usually, pulmonary hypertension in COPD is mimicked, is obtained using a combination of hypoxia, VGF inhibition, for example, multiple insult. I don't know if that answers your question.
I guess to answer the question and also plug genetic models and how they can be used with cigarette smoke. There was a paper from Robert Weissman and Cell a number of years ago where they used cigarette smoke to induce pulmonary vascular disease and pulmonary hypertension, and showed that iNOS deletion could protect mice from that phenotype. So that's you know, just a nice example of using an exposure plus a genetic mutation to really make some interesting observations about disease.
So perhaps Dr. Thatcher, whose camera is on, could go ahead and ask the question that's in the chat.
Sure. I just wanted to talk briefly or introduce and see if there was any knowledge base on, and if people are even thinking about genetic differences within the C57 Black 6 substrain. Because the Black 6 substrain at Jackson, that's the most common in the US, I think, which would be C57 Black 6A, has a missense mutation in the nicotinamide nucleotide transhydrogenase gene that affects its inflammatory responses. And I actually had a poster at ATS, I never turned it into a paper, about 10 years ago, that the Black 6J mice are more prone to inflammation from cigarette smoke on an acute exposure. So, and I don't know anything about variations between different substrains outside of the US or outside of the NJ diversity, but there are dozens of genetic differences between the Black 6 strains. And, you know, to what extent is, if you report, I'm doing Black 6, but I'm doing Black 6 and they're completely different. Well, then where did you get them from, and what's the vendor, and all of that. So does that come into play at all? Or do we just sort of think, well, smoke is more damaging than all of those other effects? I guess, how do we think about it, and what should we be doing as a community?
I think open that up to anybody who's on the call, any of the 61 participants. That's a really interesting observation.
Yeah, I mean, I think this is a cautionary tale of why, you know, everybody has seen a paper where they try to get a phenotype and then from another lab, and it's not as severe, or it's more severe. And so I think it's really important. I actually did not know about that NAD mutation. That's news to me. And I think this is a really important point to be making about why we need to be very cautious and read through those methods, and also publish our methods very, very specifically and precisely of where we obtain our mice. Thank you for making this point.
Yeah, Tom, I guess I'd like to emphasize the opposite that I've seen, and I think many of you have. When you move mice even across town between different vivariums, suddenly you get very different results that seem to be due to basically the microbiome and the cleanliness of the thing. I had a project go on. We had an EP double select and double knockout. That was a gorgeous model over at the U of M. We moved it to the VA, cheaper animal care costs and much greater cleanliness, and all the pathology went away. Over.
All right. I think, well, why don't we go to our next question it's from Lee Marsh. What is the panel's take on the combined cigarette smoke LPS model? The chronic LPS has also been shown to develop mild airspace enlargement. Why hasn't it become more commonly used?
I can answer that question. In our hands, when we use E. coli LPS, we don't see emphysema. But it can happen when we use LPS from different sources. But what we have seen is like it causes too much of an inflammation. And instead of getting inflammation like in what is observed in COPD, we get too much of neutrophil infiltration, not that many macrophages. And in addition, when we stop treating with LPS, that emphysema goes away. It resolves by itself. Whereas if you induce the emphysema with elastase, it doesn't go away. It just persists. That's what I have.
Looks like we have a comment. Oh, several comments from Ander and Francesca on that.
I just wrote that the LPS tend to induce a more airway-driven phenotype, that is inflammation and airway-driven phenotype, rather than the phenotype of COPD that we observe that encompasses vascular, parenchymal, and airway changes.
Okay. All right. Moving on. Actually, this is one of the questions, one of the pre-planned that we actually want to discuss, but how do you study the extrapulmonary consequences of COPD? This is something near and dear to my heart as well. And so I think some of the model, like I actually haven't used the smoke model, but some of the models, don't the mice have to be held immobile? So I would think that would be a confounder if you're trying to study muscle wasting or muscle loss. But this is I'd love to hear what the speakers think about looking at the extrapulmonary consequences. What would be the best model for that?
So this is also very dear to my heart. We published in 2017 and 21 the extrapulmonary causes of the cigarette smoke on the kidney and on the quadriceps. So before we published these studies, we did try LPS and elastase model, and there was absolutely no effect on the kidney and quadriceps of the mice. But only with the cigarette smoke model, and not 1 month, but at least 4 months of cigarette smoke, you started seeing, at least in the quadricep, a loss of the NADH fiber in a mitochondrial, the fiber full of the most use oxygen in the quadriceps in the smoke exposed animals. And for the kidney, you start seeing shrinking of the glomeruli after 1 month of cigarette smoke, but it becomes really overt with macroalbuminuria after 4 to 6 months of cigarette smoke. But in the quadricep, it really takes at least 3 months of exposure to get a loss of the muscle fiber. In fact, if you look at the mice that are exposed to cigarette smoke, after a couple of months, they start running less around the cage. You can tell that they're getting, and this also brings me to another question in the chat, which is the aging component of COPD. I think the cigarette smoke induced COPD is really, it induces a disease of accelerated aging that you cannot have with other models. So the smoke really shortens the telomere overall in the body of the mice. And this is why you've got accelerated aging, extrapulmonary manifestation that, to the best of my knowledge, you don't get with other, I don't know about the genetic, maybe Brad can tell about that, but you don't get with other external exposure models.
Okay. Any other thought? Okay. All right. Thank you. What role, okay, we have a question from Ander. What role do systemic inflammation on aging play in COPD-related comorbidities? So I guess we already kind of addressed the aging components. Probably the elastase model, they're probably not as aged as the other. Did the genetic models, are they typically aged? I guess that's a good question for.
Yeah, so a couple things about that. I sort of mentioned in the PIGR knockout model that there was really no phenotype too much at 2 months of age, and then it was age dependent, entirely age dependent over time. So, I think it's really important when you're using a constitutive mutation that you phenotype sort of both the initial early adulthood phenotype as well as what's going on later in life. And I guess somewhat related to that, I would point out that, probably half of patients with COPD or close to that have COPD because of problems with lung growth. And so, it could be that actually looking at developmental mutations with constitutive mutations is exactly what we need to be doing as a field to better understand, how development could go awry, and to impair peak lung growth. So I think the aging is incredibly important. It's relatively easy to do with a genetic model. You just let them sit there for longer. You got to pay for the housing costs, but you just be patient. And we did not even see an adaptive immune phenotype in the PIGR knockout model until they were a year old.
Wow, really interesting. All right, we have
And
go ahead.
Amy, you may have this as a future question, but I think if you have the money and the time, it's worth using more than one model. I've been fortunate to publish with all the speakers, but we had a study with Bradley in which we had an observation about natural killer cells activity in the smoking model that we were able to replicate without smoke in the PIGR model, a little less than a year. He sent us some cells for that. And so that makes you a little more confident that the phenomena you're studying really is robust. Over.
Oh, that was one of our questions, was if grants should propose multiple models. So no, that's really helpful. Francesca, did you want to say? Yeah.
No, I was not going to say anything, but since you mentioned the grant, so
Here, I'll go to that.
So
question.
the thing is that I don't think in a grant you should mention different model, because the grant will be that it will be too vague, I will say. But I think it depends on the question of your grant. If you want to study alveolar destruction, you can use LPS, you can use Elastase. If you wanna call it a COPD-like model, for me, I'm not gonna accept anything else but cigarette smoke. Just because if you want to call it a COPD model, it has to come from the same insult that causes COPD in humans. If you want to call it a small airway disease model, you can use whatever causes mucus metaplasia and airway remodeling. If you want to call it emphysema model, you can use other stuff. But if you want to call it a COPD model, you got to use the same stuff that causes COPD in human. And there was a question about the COPD never smoker.
Yeah,
But
that was right there.
So,
thank you.
So my question to the person who's asking the question is there really COPD, or is it just airflow limitation? I will say that is just airflow limitation, but the causes are completely different, right? So the pathology, if you look at the lung of a person with efficacious airflow limitation who has never smoked, the lung looks completely different from the lung of a person who has been exposed chronically to cigarette smoke and developed COPD. So I would assume that also the model will have to be different. So maybe a genetic model will be great for that.
I guess I wasn't sure if this question, if they were distinguishing just airflow obstruction, or if they're like, you do have some toxic exposure like air pollution. Yeah, go ahead. Okay, never mind.
Yeah, like people always say, why not all smokers develop COPD? So there must be other factors that actually induce COPD, right? So there may be cold, they may be exposed to air particulate material that comes out of the, exhaust, or coal miners, or people who are exposed to biomass, fumes from biomass fuel. So the COPD can be induced other than cigarette smoke as well. But
So I'm sorry, go ahead.
In the grant, usually they like cigarette smoke model, but at the same time, reviewers also ask, oh, okay, it doesn't cause anything until 6 months, and then it goes this whatever the cigarette smoke, the changes that are induced by cigarette smoke will go away. It gets resolved. You stop smoking the mice, it will resolve. So reviewers always have questions. So always we mention that what question you're asking and why you're using that model. That will help.
So Francie, I wanted to give you a chance to respond because and I adore the fact that you're not pulling any punches. You're really stating your opinions here. But I guess I was going to ask kind of the converse of Uma's thought is a fundamental problem with the mice, besides anatomy, is just once you have a stimulus, any of these stimuli, they all get it. And that clearly is not what human COPD, we don't have a way yet to get the heterogeneity of susceptibility. Do you or anyone on the call have an idea about that? I mean, again, if all the money in the world were possible, which it clearly is not right now, we probably should be doing what David Massapros did, outbred mice that have a wild gut microbiome, and you would expose hundreds and hundreds of mice and do multivariate regression on them, but that's clearly not going to happen. Over.
Also, all the mice we study are all the same genetic background.
Exactly.
So that is a major flaw of our mouse model, but I think it's as good as it gets. I wanted to say something about what Uma said. So if you expose the mice to 6 months, 4 to 6 months of cigarette smoking and they get emphysema, small level remodeling, that stays. Even if you stop smoking the mice, and we have done it extensively for 1 entire year, the remodeling and the emphysema stays. The only thing that goes away is the inflammation. So the chronic pathology, and if you measure the flex event and the pulmonary function to mice that have stopped smoking 1 year before, they still have increasing compliance, increasing elastanes. So the only changes that go away are the acute changes, which is mucous metaplasia and inflammation. All the other changes stay. And look, I agree that not everybody can afford the smoke model. I also would like to study many other things, but I cannot afford them, so I don't study them. But again, you can still study pathology to COPD as long as you call them with the right name. You label them correctly.
I think every speaker is kind of agreeing that it's really about the question, that we have to kind of make sure we know exactly what we're proposing. Were you going to say something, Dr. Curtis?
No. Turn the camera back on because I stopped coughing. Go ahead.
Okay. All right. Okay, so just lots of comments. I love the enthusiasm from the audience. Talked about immobilizing a mouse, an old model of stress.
There is one comment on alpha-1 antitrypsin.
Oh, where? Okay. Where is it? Oh, please go if you can respond. I don't see it.
Yeah, hi, Karina. So alpha-1 antitrypsin is, I will consider, I don't know, Brad, what do you think about that? I will consider a genetic model, right? It's not different from other genetic models. And I still, in humans, even if you have alpha-1 antitrypsin, you still need a second hit to develop emphysema. So in mice, I would assume it's the same. I mean, I haven't worked with alpha-1 antitrypsin. Maybe Brad can say something about alpha-1 antitrypsin deficiency mice.
Yeah, so this was really a tour de force effort because mice have 4 different copies of the gene. And so that required knockout of all 4 versions to get a phenotype. I can't recall the paper, but it's in my second slide. I put that in that list of models. And they do get disease. I believe that it had to be induced by cigarette smoke. And Francine's absolutely correct that typically even in in ZZ mutations, most of those patients have to smoke to get disease. But it is an available model. It just came out a couple years ago, and a real technical tour de force to get that.
And by the way, you don't need cigarette smoke. I mean, Uma is correct. COPD now is caused by second-hand smoke exposure. It can be like particulate matter 2.5 is now the main cause of COPD worldwide, more than cigarette smoke. So certainly you can expose the mice to different pollutant, but it has to come from your nostrils. That's my take.
All right. Well, I think we're actually at the end of the hour. I really wanted to, we covered, I think, almost all the audience responses, which is great. But I really wanted to thank our speakers so much. This was wonderful to get everyone talking about it and the audience too. So thank you so much, Dr. Polverino, Sajjan, and Richmond. Thank you, Dr. Curtis, for helping moderate and develop this session. I really appreciate so much your involvement. And
ATS for sponsoring.
And ATS for sponsoring.
Thank you.
Thank you so much, everyone.
Thank you.